Oh. Yeah. Okay, sure.
All right, we're going to continue on here with the cortex, the cerebral cortex, the gray matter. So it's useful to know that, well, first of all, let's look at this picture there. Gray matter of the cerebral cortex. It helps to know that different regions of gray matter have different functions. The gray matter, again, like I said before the break, the brain is arranged in the gyri and sulci, and only the thin few millimeters on the outside is the gray matter. All the white matter is the inner tissue. We'll talk about some white matter nuclei next. Again, to review, the brain is color-coded there so we can easily recognize the different parts of the cerebral cortex. You got the 4 lobes that we talked about. That would be like the anatomical arrangement, the 4 lobes of the brain. But here is kind of showing you the functional arrangement. It's basically going to be motor, sensory, or association areas. Okay, I'll define what that means in a second. But let's first review the 4 lobes of the brain that we talked about before the break. Parietal, this light blue right here. Posterior part of the brain is the occipital lobe, temporal lobe, and then the frontal lobe. And those different lobes, the only gyri and sulci you got to know is shown right here. Let's remember that the central sulcus, right down the middle right there, be able to identify that. Then be able to identify the pre-central gyrus in front of it and the post-central gyrus posterior to it. Which one has a sensory function? Post. And the pre, that's the motor cortex. Okay. All right, so I want to talk about other motor areas of the brain. So motor, think you're commanding your body to do something. The primary motor cortex, that's skeletal muscle, voluntary movement. Skeletal muscle. That's, well, in another lecture we called it, somatic motor function, right? The motor neurons. The premotor cortex, that lighter red, just anterior to the primary motor cortex. Oh, I misspelled motor. Okay, what's wrong with me? Sorry, motor. Premotor cortex. Now this cortex is like a staging area for planning movements. A staging area. For coordinating basic movements in into more complex ones. Kind of like the thinking that has to go through, the examples are given, like, for example, typing. Now, a keystroke is a very basic function, just like press down. But I don't know if you remember learning how to type. It takes a long time where the home keys are and typing many words per minute. It's very complex. You take basic movements and learn in the learning process, they become more complex one. Another common example is playing a musical instrument, you know, maybe a piano, okay? So those kinds of things. It kind of stages the area for coordinating these basic movements into more complex one. Frontal eye field, moving your eyeballs. Simple enough. Not vision, moving your eyeballs, looking down, looking up. If I want to look down and to the right, this eyeball has to look down and laterally. The other eyeball has to look down and medially. Okay, I mean, you don't think about these things, you just do it. You know, you just move your eyeballs and you take it for granted. You have a part of the brain that allows you to move your eyeballs, makes it easy for you. Broca's area. It's for motor speech. Just the act of me speaking out loud to you is motor speech. What's the motor part of speech? The words have to come out of my mouth. So I have to like move my lips, tongue, and palate to form and shape the words in a way that you can understand. Okay? Again, something you take for granted. You can tell there's a whole area of speech pathology for individuals, maybe due to stroke or developmental disorders or any kind of a thing where speech is affected. Broca's area, prefrontal cortex. So I want to put a line because prefrontal. It's not strictly a motor thing. It's more for deep thinking. I'll just put deep thoughts. Okay. I want to move on. Talk about some basic sensory functions of the brain. Do you ever wonder why when you think really hard, you furrow your brow or you touch your head? I mean, that's the part of the brain that's kind of doing the thinking for you. But it's not really a motor function, but it's kind of close to one. Okay, these are strictly sensory functions. Sensory areas. the cerebral cortex. Well, the one we learned before the break is the primary somatosensory cortex, or the postcentral gyrus. Okay, so basically, this is where you feel your body. Every square millimeter of skin can be felt, okay? And all the information gets sent to here. The skin is the largest sense organ of the body, the sense of touch. Basically, feel your whole body. Taste is kind of between this little area here. The word for taste in anatomy is gustation. Taste your food. And when I start working on the cranial nerves on Friday, I'll teach you the cranial nerves responsible for taste. Okay. Wernicke's area. Put it in parentheses, it's not strictly a sensory function. This is the area for language comprehension. You know, like when you get an instruction manual, I hate instruction manuals. It comes in 7 languages, and I keep just flipping till I find one that I recognize, English. Language comprehension. Okay? This is not motor speech. Reading out loud from a book is both language comprehension first, motor speech second. If I'm reading, I have to be able to recognize the word first. That's this. That's comprehension. But me vocalizing the word out loud is motor speech. I guess that comes second. Okay, this is for language comprehension. And moving on, let's see, okay, vision and hearing, that's pretty basic. Vision is in the occipital lobe, hearing is somewhere in the temporal lobe. If you receive a blow to the back of the head and you see stars, so to speak, it's because the occipital lobe is back there. Appreciate your eyeballs are in the front. They got to go all, the signal optic tracts got to go all the way to the back of your brain for vision. Okay, hearing. We'll learn about the inner ear structure. That's like one of the last lectures. I'll say it receives inputs from the inner ear structure. Just to give you a little preview on that lecture, it's all about the hair cell. All about the hair cells in the inner ear. Talk about those later. But anyways, that's the part of the brain for hearing. Let's see here. And so I don't know if you've noticed this, but the association areas, for example, vision is a good one. Here's vision. The dark blue represents the primary area for vision. But all the cortex close to it, in proximity to it, colored in lighter blue, is the association area for vision. Another example. The dark blue here is the primary area for hearing, but this area around it is the association area for hearing. Association areas kind of make sense of the world for us. they take all the incoming information and they put the world into context for you. I'll just kind of read what I have on my slide here. There are large areas of cortex. They lie outside the primary areas, but in close proximity. They integrate diverse information into purposeful action. So I have a PET scan here. The areas that light up mean is where the most intense activity is occurring. For example, if you see a red stoplight and you're driving a car, that sets off this part of the brain, the occipital lobe, and its association area. However, you must make sense of that. So what does it mean? Oh, I hit the brakes. Obviously, if you know how to drive, you know how to do that. Do you hit the brakes every time you see any red light? No. Only this one on a traffic stoplight. If you're not driving the car and you're sitting in the back seat, do you hit the brakes? No, the driver does. I mean, you know, this is like a good example of you know what to do when your brain lights up. The association areas give you purposeful action. What happens if you're distracted? You run the red light, right? So I mean, the association areas are very important to us. The other concept we like to teach students is the idea of homunculus. Disproportioned person shown right here. Homunculus represents the amount of cortex devoted to a body region for touching and for motor command. For example, for feeling, look at homunculus. Based on the size of the body parts, which are the more sensitive parts of your body? Yeah, your hands, your lips, you know, very sensitive parts of the body. And so the topographical map of homunculus is laid out on both the motor and the sensory cortex, right there. Okay, this is a frontal section view. So it's been all mapped out. We're able to know this because you could do studies. You can expose the brain. The patient can be conscious because the brain has no pain receptors. You can stimulate parts of the brain and ask what they're feeling, or you can see what they're doing. And so this has all been mapped out. The cerebral cortex is basically a topographical map. Well, anyways, we're going to move away from the outer gray area, gray cortex, to the inner white matter. And the only white matter I want you to know is the basal ganglia, or basal nuclei as they're sometimes called. Everyone knows what a cell nucleus is. We already had the cell biology chapter. But in neurophys, a nuclei is a cluster of neurons, a cluster of cell bodies. Okay. So that was the gray matter, white matter of the cerebrum. Study the basal ganglia. Basal ganglia are also important in basic motor function and moving your body parts. It works in conjunction with other parts of the brain. for, say, starting and stopping, basic movements. And it allows to like make adjustments to a movement. Kind of like, you ever like grope in the dark for a light switch, and you do it based on feel. You can't see too well, you just kind of groping around. Oh wait, that's the edge of the door. I've got to go back a little bit. The feel, the touch, and different things. Well, eventually you find the light switch, and these kind of nuclei help you function in that capacity. So I kind of have to map it out. So let me kind of blank the screen for a little bit and map it out for you. For the basal ganglia to work, it needs to receive inputs from 4 regions of your cerebrum. So let me draw the brain here. And let me draw more or less where the central sulcus would be, the black squiggly line. Let me draw the sulcus right next to it there. Let me draw it right there too. Let's look at, well, you tell me. I'll color this blue. I'm going to color the thing in front of it red. What did I color red? What do you think I'm going for? You should be thinking motor cortex or pre-central gyrus. So then what's the blue? Post-central gyrus for sensation. Their primary somatosensory cortex. Those are 2 regions that work with basal ganglia. There are 2 more. The association areas for motor, let me just kind of draw it like that. And also the association area for sensory. Let me kind of just draw like this. So let's just kind of number those 4 regions, 1, 2, 3, and 4. 1, 2, 3, 4. So in case you forget when you look at your notes tomorrow or something, 1 is, it's the association area for the motor cortex. So then 2 is the primary motor cortex. 3 is the primary sensory cortex, sometimes called somatosensory, but I'll just say sensory for shortness. Sensory cortex. And its association area, which is basically number 4. Primary sensory cortex, association area. Okay, some areas of the basal ganglia, there are three things. They include, they're inside the brain, but just for a diagram purpose, I'll draw it outside the brain here. There's a structure called the caudate, and another structure called, okay, that's C for caudate. So this is the basal ganglia. That work with 1, 2, 3, 4. So the C is for caudate. And then there's putamen, P, and GP for globus pallidus. Those are the other parts of the basal ganglia. So P is putamen. GP, globus pallidus. Sometimes they refer to caudate and putamen as striatum. That's another term I've seen there. Put that in there. Also called striatum. All right, so how this works is, if you're gonna make adjustments to movements, start and stop movements, everything, you need to receive inputs from 1, 2, 3, and 4. So what happens is, let's list this in steps. Step number 1, for the basal ganglia to work, projections from one, 2, 3, 4. Those 4 things I define here. The projections are called corticostriate fibers. These corticostriate fibers project to the striatum. Again, that's the C and the P, caudate, the putamen. So I'm just going to draw lines that go from all 4 regions to caudate, putamen. Call it 1, 2, 3, and 4. So all of these we're going to project to both the caudate and the putamen. So both to here and to here. So that's what I'm drawing there. So in my diagram, these 4 lines represent my cortical striate fibers. And think about what's going on here. You have 2 little spots in the brain, the striatum, that's receiving all the motor and all the sensory input. Okay. So from there, call it step number 2, the striatum project to the globus pallidus. GP. So I'll just draw a cell projecting to there, another cell projecting to there. I'll draw another cell body there. Step number 3, the globus pallidus projects to thalamus. So I'll use another spot on the board to illustrate the thalamus. Remember, the thalamus is composed of those neurons. It's a switchboard. Well, anyways, some of the inputs from the globus pallidus will project to the thalamus. And lastly, the thalamus will project back to number 1. To number 1 on our figure there, the premotor cortex. So that's the circuit. You receive all the inputs, motor and sensory, and if you have to make an adjustment in your movement, you project back to the premotor cortex, and then you can adjust your movement. So I guess as a last step, I'll put a cell that goes down from the primary motor cortex that projects down to spinal cord. And we can list that as a 5th step to make an adjustment on the movement. Number 5. The primary motor cortex, I'll just put motor cortex, projects to spinal cord. Like, for example, if I'm washing dishes and I have soapy hands and I grab a wine glass with my soapy hand and it begins to slip out and I feel that and I adjust my grip so I don't drop the glass on the floor and it breaks. That all happens in a split second. That's all this. You make adjustments. Like, oh, that reaction time. If you ever observe someone with Parkinson's or some other akinesia disorder, and they have tremors and things like that, that's a dysfunction. I didn't get into too much detail. I don't really want to go there, but that's the kind of thing I'm talking about, where you have feedback mechanism to adjust movements. So this is the diagram. Let me just describe it more here. The basal nuclei, or basal ganglia, those are these 3 things. What they can do, they can compare motor commands for movements with proprioceptive feedback, okay, during an evolving movement. I gave the example of grabbing a wine glass and it almost slips out of my hand. How do I know it slips out of my hand? You can feel that, right? It can do this because it has inputs from both motor and sensory fields. So as you feel the glass slipping out of your hand, you adjust your grip. Sometimes you drop the glass, sometimes you don't. Here's an anatomical picture of how they fit in the brain, because I kind of drew it all over the place just on the chalkboard so I can make a diagram out of it. But they're in the white matter. The color purple here. Here's caudate, right next to the ventricle of the brain. Caudate, putamen, globus pallidus. Okay. Basically it. If that's interesting to you, you could take a neuroanatomy class and the entire semester, all you do is learn about those different regions of the brain and different things that they do. When you do brain in a day, which is what this is, we really don't have time to cover too much else. All right, so what I was saying is this constant comparison allows you to adjust muscle tone during voluntary movements. It may also control during internally generated movements, such as walking. Some of the other things we like to address are the language aphasias, okay? Because language is so particular to humans, we bring it up in the brain lecture. An aphasia is an inability to speak in one way, okay, or another, and it has to do with the brain. The brain and behavior, you can buy a big textbook on it. I'm just talking about language. And the areas of the brain we talked about were Wernicke and Broca's area, and how they studied the brain back then is you can only study the brains post-mortem. And they would like know the patients, and when they died, they would examine the brain, and they would kind of see what was wrong. And if they found lesions in the brain in those areas, and they can make the connection with the language aphasia they observed in their patient, you can begin to postulate what that part of the brain does. In particular for language, I want you to know what an aphasia looks like if there's a motor speech problem or a language comprehension problem. Okay? So language and speech depend on motor areas in occipital and temporal lobes. So this is basically reading out loud. So how it works is, as you all know, you can all read. You see the word, so the visual cortex lights up. Then information is communicated to Wernicke's area. Do you comprehend the words you're reading? Okay, in a class like this, maybe you don't understand all the words, but anyways, that's the area of language comprehension. You understand the word. Then, if you're reading out loud or talking out loud, that's the area for motor speech. You want to execute the function to mouth the words, to vocalize the words. Okay? So that's why after motor speech, you have the primary motor cortex light up. Okay, so that's in sequential order. This is just showing you the communication of fibers from Wernicke's, yellow, Broca's, red. There's a connection. If you first understand the language, then you can, it helps your brain say it out loud. Okay. All right. So damage to Broca's area, I call it Broca's aphasia. Aphasia. Broca's aphasia. Motor speech is affected. Speech is, it's hard to get the words out. It's hard to talk for this person. Hard to get the words out. Simply put, speech is effortful. That's what we call it. So for example, you'll omit words. Like if you want crackers, you'll just say crackers. You don't say I want crackers, it's too hard. Okay, and so you drop words of a sentence, and you hope context you can communicate what you're trying to say. The other kind of speech, which is more fluent, but still there's a deficit, is Wernicke's aphasia, sometimes called fluent aphasia. Fluent because you can speak, no problem, but you're speaking gibberish. They kind of call it a word salad. Okay. So I'll say speech is not effortful. Speech is fluent gibberish. It just doesn't make sense. They call it word salad. It's like all the words are mixed up in a salad. The best way to appreciate what these aphasias are is just to kind of like look at the YouTube clips. So I got them all queued up here.
Are they captioned?
I'll see if there's captions. Yeah. think they might be captioned.
Okay. So what?
This is Sarah Scott. She's the first case I want you to study. She's an example of Broca's aphasia. Be very observant. Look at everything she's doing to help herself speak.
Can you click the captioning button again? It has to be red.
Okay,
Thank you.
you are welcome.
What's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
You're 19. 19. Yeah. And what happened to you?
Stroke.
You had a stroke last year.
Yes.
And what happened? Can you remember what happened?
School and English class,
Okay.
and I book and I read it aloud, but I can't because stroke, and so I turn and also it's the same as kind of thing as you know.
Pins and needles.
Yeah,
like pins
the
and
same, and also arm as well.
leg.
Yeah, yeah.
And what happened after the stroke with your speech?
But I can't.
You have speech problems.
Yeah.
Do you know what that's called?
Yeah. Aphasia.
Yeah, good, good. I haven't heard you say the word before.
I write it down.
And that helps?
Yeah.
Okay. So you are going to go to university. What are you doing now?
But
What do you do now? Every day?
I will speech the speech.
Therapy?
Yeah.
Okay.
And also riding.
Riding what?
Horses.
Yeah, good.
And connect.
Connect, yeah. You like Connect, don't you?
And it, yeah.
Why do you like Connect?
Aphasia, so I can the same kind of problems and speech.
Okay. And how do you feel?
I'm fine.
You're fine.
All right, so I'll pause it there. I think you get the idea. So do you see why I gave these notes? Do you see how hard it's difficult for her to speak? And why is she writing things down? What does that do? Somehow it helps her because she has no problem with writing. Somehow writing it makes her brain, you can see it, maybe it helps her say the word. It's very curious, okay? And students always ask me, well, can you get better? And the answer is yes, with therapy and hard work. And I want to show you a clip 6 years later. This is 1 year after her stroke. Hold on, let me cue it up. It's 6 years. That was 1 year. This is 6 years later. Sarah Scott. Ads on YouTube. Skip ads. So see if you notice a difference 6 years later as opposed to 1 year later.
It was my stroke anniversary last week. So I had my stroke 6 years ago, which is a long time. It seems like yesterday. But yeah, so my age now is 24, and I'm still working at the school. My job is science technician. It's not full time, but it's a job that I love, and I really, it's a good, fun job. It's just things that I can do. It's very what's the word? Practical stuff. So even though I need to talk, it's more things that I can do after my stroke, so that's good. And we're still doing our stroke group, and that it's good that it's still going. But there's things that I want to do, some goals that I want to do, as maybe still working at the school, but maybe doing another job as well. I was thinking about doing a jewellery, making jewellery.
Okay, so just playing for less than 2 minutes, I think it's pretty clear that there's improvements. Wouldn't you agree?
Yes.
She's not dropping as much words. I still think speech is effortful. And you also look at her life. The quality of life is really good. She's trying to meet with people to talk about it. She's trying to hold down a job, thinking about other jobs as all young people do. So that's a great example of Broca's aphasia and how you can improve. The other case, I'm going to show you Wernicke's aphasia. You'll see the difference. It's very obvious. Word salad.
Are these both issues in the frontal lobe?
Broca's is frontal. Wernicke's is more parietal.
Okay.
Hi Byron, how are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people over them over there. They're diving for them at the moment. But they'll save in the moment. He'll water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
juvenile.
sort right here and they'll save their hands right there for
And-
them.
And what were we just doing with the iPad?
Right at the moment they ain't gonna show a darn thing.
With the iPad that we were doing. We, like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're all with them. Other people are working with them. I'm very happy with them.
Good.
This girl was fairly good and happy. And I play golf and hit other trees. We play out with the hands. We save a lot of hands on hold for us, other hands. I don't know what you get, but I talk with a lot of hand for him. Sometime. Am I talk of any more to say?
All right, thank you very much.
Thank you very much. I appreciate it, and I hope.
Okay, that's Byron there. That's a good case study of word salad. Wernicke's aphasia. So any comments or questions on Wernicke's aphasia? The case study says it all. Could you understand him? You could understand what he was saying, but was he making sense? Well, no, that's the whole point. I have some YouTube links there if you want to look at those later. He had no problem speaking. That's why we say fluent. But the words are all mixed up. And she's asking him one thing, he says completely something else. And it's very interesting. You don't know if he understands what he's saying is gibberish. To him, he's probably making perfect sense, but you really don't know. You can't get inside his head. But that's when you have that's what it looks like when you have a dysfunction or a problem with Wernicke's area. The last case I wanted to study is split brain. Or the corpus callosum is severed. So the right brain and the left brain cannot communicate. So let me kind of show you what you're going to see in this video. Let me show you this diagram here. What we're going to see is they operate independently, but they need to talk to each other under normal circumstances. But when they can't talk to each other, well, here's what you first need to understand. In the world you see, here's the binocular vision you have. You can divide it into right and left visual fields. And the color code makes it easy to understand. Left in blue, right in yellow, where it's kind of sketched out there, that's kind of more like a peripheral vision. Here are your eyeballs. The back of your eyeballs send cranial nerve 1, optic nerve. The nerve fibers send information from both visual fields. For example, the left eyeball has information from both right and left visual fields, correct? However, both eyeballs, notice how the information is sent. All the information from the right visual field, if you were to gaze at a point and fixate on it, all the information to the right will go to the left brain and vice versa. All the visual field from the left visual field will go to the right brain. So you see the left visual field with your right brain and vice versa. Okay, so earlier I said, I talked about cerebral dominance. Left brain is for language, the right brain is mute, can't talk. So in a test for a normal person, if you show them a picture, they're gazing forward, but you show them a picture of a saw, okay? Well, all the information, because it's in the right visual field, will go to the left brain. Because the left brain can talk, when you ask the person, what do you see? They'll say, oh, saw. However, if you put the information in their left field of vision, remember they're always gazing forward at a point, the information goes to the right brain, which can't talk. But that information is sent to the left brain, you ask the person, they say saw. However, if the corpus callosum is severed and there's no communication, the two are operating independent of each other. If you put it in there and you ask them, what do you see? Then they can't answer you. They can't say they see it, even though they do. It's very odd. Also remember that your right brain controls left body and vice versa. Left brain controls right body. So it's the right brain that saw it. So what you can do, this is really bizarre. You can ask them, okay, you say you don't know. ask them to draw it with their left hand, and they can draw it. They'll draw a saw with their left hand because the right brain saw. It's very weird. And when I first read about this in my neurophys books, I didn't even believe it until the magic of YouTube. I actually saw the case. I was like, oh my God, that's so bizarre. I'm going to show it to you. It wouldn't take too long. It's a great example of how we all have two brains that operate as one. This guy, you're going to see how it's like you have two brains and two different personalities in there. Yeah, I don't think this one has captions. It's an older video.
Left hemisphere and right hemisphere now are working independent of each other, but you don't notice it. Now you just kind of adapt to it. It doesn't. You don't have any feeling different than it did before.
7 years ago, Joe had brain surgery to allay the effects of severe epilepsy. His surgeon cut the nerve fibers connecting his left hemisphere with his right. While the operation was a complete success, Joe's unusual case offers an extraordinary insight into the machinery of mind. This fiber system, the corpus callosum, is located midway between the two hemispheres. When it was surgically severed in Joe's brain, the transmission of information between the two hemispheres was halted. Michael Gazanica.
What we can do is play tricks by putting information into his disconnected, mute, non-talking right hemisphere and watch it produce behaviors. And out of that, we can really see that there is, in fact, a reason to believe that there's all kinds of complex processes going on outside of his conscious awareness of his left half brain. Joe, I'm going to show you some things. I just want you to tell me what you see. And here we go. You ready? Look right at the dot.
Okay.
Right. Okay, you ready? Right at the dot.
Grapes.
Good. When Joe focuses on a point, right at the dot, everything to the right of the point goes to his left brain, the dominant hemisphere for language and speech. Right at the dot. So we can see here that when we flash a word or a picture, Joe is easily able to name it.
See it?
Close your eyes and let your left hand do a little work here. Okay, what do you got there?
Pen.
Okay, very good. Now, when a word or a picture falls to the left of a fixation point, that information goes to his disconnected right half brain. And as we can see here, Joe is unable to name it. Joe is able to draw the picture with his left hand, the left hand getting its major control from the right half brain. What do you draw?
Okay.
What do you see?
Wheel on one side and on the other side.
So even though he can't name it, his left hand is able to draw out the picture of the stimulus of the picture or word that we presented to his right half brain. What did you see? So just close your eyes and draw with your left hand. Just let it go. That's nice. What's that?
Saw.
Yeah. What'd you see?
Hammer.
What'd you draw that for?
I don't know.
what we have with Joe is just a dramatic example of a neurologic case that really allows you this window into the non-conscious and how powerful non-conscious processes are at influencing our conscious self, our personal self. What Joe and patients like him, and there are many of them, teaches us is that the mind is made up of a constellation of independent, semi-independent agents, and that these agents, these processes can carry on a vast number of activities outside of our conscious awareness.
Okay. I think you get the point. Now, that kind of shows Joe's deficits, right? There's actually also advantages to having two brains that work independent of each other. And I'm going to show you just one more brief clip. The challenge is you put your hands in a box and you're asked to draw two different objects, one with each hand, simultaneously. Normal people can't do it because it's like these don't work independently, they work together. When you force them to work independently, it's very difficult. Okay, what you see is Joe can do it a lickety split. It's very, very interesting. It'll be the last thing to watch. Hold on. Alan Alda is the kind of the guest speaker in this documentary.
Well, we're seeing the fact that Alan's hemispheres are connected.
Hold on a second. Just a little bit.
I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we're seeing the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside-down duck.
Okay.
But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees. Wow, look at that. It's really like two different people doing the same.
That's right. That's the idea.
Okay, Joe, I want you to keep your hands up.
In an experiment that's now a classic in brain research.
All right, that's the one thing I wanted to show you. So I thought it was really cool. Anyways, that's the Brain in a Day lecture. Based on the time, I'm just going to dismiss class at this point. But the one thing I would like to do is to remind you of your lab quiz on Friday. On what? Skull bones. Okay, and you've been studying for that, I hope, and you got the pictures, right?
Yeah.
I do have your lab practicals. If you would like those back, just come up here. If not, you're dismissed. I'll see you Friday. We're going to do the groups again. Group A first, Group B to follow. Yeah.
couldn't figure out what it was. We went to the next one, and then I was able to see it. That makes sense. I wasn't sure on this. And then when we moved to the next leg, he had it all together. And I was like, oh, that's where I was doing it. Can I squeeze behind you?
Is that fine?
I can put that there. We'll take care of it now, so next semester we'll make sure that it's not just students, but it should be good.
Thank you.
Alveolar process. Yeah. That's just parietal bone, and that's got to be something that. Are you sure they're not in the book?
Yeah, they're not.
We'll check that. So, I mean, you
Yeah, I started studying last night, and I have.
So, look, if I would have done better, look, I erased that underneath that says soleus.
Oh.
So see, I had the right one,
Yeah,
and
I
I put the wrong. And then I did the same with this one.
Somebody moved.
So how do you like, not second guess yourself, like, just go with your gut. Doing that.
Yours is right.
just don't change your answer. That's how.
I
That's
get
the
so
easiest
mad
answer.
afterwards though, because I'm like, I had it and I changed
I know. That's usually what happens, so I'll advise you not to change your answer. Yeah. You're right, yeah, these leader lines aren't there. I don't know why. Maybe this is from an older edition.
Okay, and then I saw there was like 2 other ones. Yeah, where is that? That was not on.
Oh, I'm not going to ask you that.
Oh, you're not gonna ask
That's
me?
just tea. But I'm not going to
Okay,
ask
I
you.
thought there was a specific. And then.
So what part?
And then for this one also, you were pointing towards here, but it's not labeled for that either. So there's
Oh, yeah. Actually, that one you do need to know. That's the mandibular fossa.
mandibular.
It's labeled on the other side.
Oh, okay. It says mandibular foramen.
Oh, I'm sorry. Mandibular foramen.
It's foramen.
Yes, it's foramen.
Is this how you practice?
Yeah, that's with the,
So
I'm in
Sarah told
SLC and she told me to do this. I was like, oh my god, that's so smart.
They do release.
Professor?
Yeah.
I think we should take 1 point because I say I'm Dr. Burkle, but I say I'm not.
Oh. Oh. Dr. what? For Morris? Oh, okay. Yeah. Okay.
Yeah?
So no problem. Yeah, let me read it. I'll get back to you later. Anything else?
No, no, no. Thank you.
I have a question about the lecture. Sam.
Yeah.
Can you tell me what's great, like what do you usually get?
I don't know. I'd have to look it up from last year. If you want to email me, I can look on my Canvas and email you back with
I'd
that.
like it if you could just give me any advice on how to study that, because I have moved over.
Oh, yeah. Well,
the
I would
study
rewrite.
guide,
I would
and it
rewrite.
does look like it's a lot to.
I would rewrite your lecture notes, and then after you study your lecture notes and you understand your lecture notes, try to answer the questions from the study guide for that lecture. That's what I would do. Use the study guide as a review.
And then for the final, the study guide you gave me the final, is that what's gonna be on the final?
Yes.
Okay, alright.
Yes.
Thank you.
So that part's simple.